So good afternoon, everyone. I'd like to welcome you to the second webinar of the JoinHealth Spring Series. The JoinHealth webinars are a collaborative initiative from the Health Cluster Portugal and the IT Health. Miguel Amador is here today in representation of the IT Health stars in Portugal. These webinars have been planned with the aim to promote networking in an open and informal environment. So please feel free to ask questions and to participate in the discussion using the Q&A box of the Zoom. And today's webinar will be about preclinical research. Today's agenda, we will have Dr. Anna Olsen, who will approach the topic of non-animal models in biomedical research, and Dr. Joslyn Dimaggio to present the congenital research infrastructure on non-human animal models. So thank you so much, both of you, for kindly accepting our invitation. After these two presentations, we'll have sort of a roundtable to discuss a bit about the future of preclinical research and global trends. And again, I invite you, all the participants, to join this discussion and drop your questions in the Q&A box. Before we start the actual presentations, just a brief note about the presentation slides. They will be available upon request. We'll have this satisfaction survey in the end of the webinar, and you'll be able to ask for these presentations there. Also, the recordings of this session will be available on the YouTube channel of the Health Cluster Portugal in a couple of days from now, so you'll have a recap of this session. But now, without any further delays, we'll move to the first presentation by Dr. Anna Olsen. She's a researcher at the I3S at the University of Porto, where she leads a research group in laboratory animal science. Her main research interests are animal behavior, welfare, and ethics in the use of animals in research. And she has been coordinating several European projects on these topics. She chairs the I3S Animal Welfare and Ethics Committee, where she's also responsible for the training of researchers in laboratory animal sciences. She's on the scientific board of the Swiss 3R Competence Center. She's a trustee of the University's Federation for Animals Welfare, and she also teaches in doctoral programs at the University of Porto. So thank you so much, Dr. Anna Olsen, for being with us here today. And I invite you to share your screen.
Thank you very much. Let me just get the presentation started. Okay. Thank you very much for the introduction and for the opportunity to contribute to this webinar. Non-animal models in biomedical research is a somewhat unusual topic for me. I think you can read between the lines of the presentation that I work a lot closer to the animal models, but I have a particular reason to be interested in the non-animal models, and that has to do with the ethical aspects of animal research. My focus today will be on strategy, policy, and training, also for reasons that will become evident during the presentation. And I will start in what is perhaps an unusual place to start a talk in the context of science, which is by citing a piece of legislation. But the Directive 2010/63/EU on the protection of animals used for scientific and other experimental purposes is really a piece of legislation that I recommend anyone that is professionally, minimally close to this topic to actually read. If nothing else, I recommend you read the recitals, which is the first part of a directive. It's the part that doesn't give the norms. These are the articles that come later, but the part that gives the reason for having this legislation and the reason why this legislation looks the way it does. And I'm going to cite here recital 10, which tells us that the use of live animals continues to be necessary to protect human and animal health and the environment. But it also tells us that the final goal should be the full replacement of procedures on live animals as soon as it is scientifically possible to do so. When I say that it is necessary and the goal should be, I'm meaning that this is what the European Commission, what our legislative text in this part of the world where we are working at the moment, is making these claims. At the moment, it is necessary to continue to use animals, but it also should be the ambition to work towards a total replacement. This is not something that the European Commission invented 10 years ago. These arguments are much older than that. And to a great extent, they are based on something which is known as the 3Rs principle. And the 3Rs stand for replacement of animal experiments with alternative methods, reduction of the animals used, and refinement of the methods to minimize animal suffering. This principle was presented by these two British gentlemen who, when they wrote the book, were much younger than on this picture, because the book, The Principles of Humane Experimental Technique, where this principle is first published, came out over 60 years ago in 1959. It is really impressive how Russell and Birch were really ahead of their times in formulating this principle and the text around it. It took a long time, several decades before this principle really came to penetrate the way we speak about working with animals. But it is still as relevant as it was 60 years ago. And this is to illustrate how long it took for this really to capture attention. These are citations to the book as identified in database searches. And you see that it's only in the 1990s that they started to find citations of any number, sizable number, and it's really only into the 2000s, so into the present millennium, before the numbers really start to be anything worth considering. And as I think is obvious from the context, my focus in this talk is going to be on replacement, whereas usually I work mainly with refinement. But the focus here will be in replacement. And if we look historically at replacement, we will find that this effort have been much greater and have been during a long period predominantly in one area of animal use, which is toxicology and safety testing. So there have been consistent efforts in developing non-animal alternative procedures for testing chemicals and cosmetics, pharmaceuticals and other products from the 1970s. And we have a number of collective entities appearing. By this order is chronological. So we have the FRAME, which is a foundation which is based in the UK. We have a Center for Alternatives to Animal Testing that started in the US, but is now also represented in Europe. Then we have the German ZEBET, which is the Center for Documentation and Evaluation of Alternative Methods to Animal Experiments. And the latest to appear here is the ECVAM, the European Center for the Validation of Alternative Methods. This, on the other hand, as being part of the Joint Research Center of the European Commission, is the strongest player, the most heavyweight player in this context. A lot of this work has been driven obviously by a strong wish to get away from the use of animals, and it's been connected with political will and lawmaking, and especially in the context of animal testing for cosmetics. Cosmetics, for obvious reason, became a kind of a poster child in this context, with manifestations against the use of animals, and the rabbit is really the prime poster example here. Anyone who thinks critically about whether or not it is justifiable to use animals will find it very difficult to justify the use of animals for something that we don't really need, meaning cosmetics. So there have been, for a long time, the position on the European level has been that animal use for cosmetics testing is only accepted if there is no alternative. And coupled with this, there has been a strive to develop alternatives, and gradually, as alternatives have been developed and have been validated, the corresponding animal test has been banned. This has been a long process. You see, this goes from 1993 to 2013. In 2013, finally, all the tests that had been in practical use for cosmetics testing had found a validated non-animal alternative, and it was possible finally to completely ban the use of animals in the testing of cosmetics, both ingredients for cosmetics and final products within the common market of the European Union. This talk is not about cosmetics testing, but I'm using this as an example and an illustration of how this really strong ambition in legislation also helped and drove a technical development. In this context, we've also seen the appearance of several more entities. We have the EU reference laboratory, that is what used to be the ECVAM. And we also have the European Partnership for Alternative Approaches to Animal Testing, which is a voluntary collaboration between the European Commission, European trade associations, and companies from different industry sectors. And here we've got the pharmaceutical industry as well. So here we are in a wider context of activities. Another example of a political statement here is the Dutch ambition to completely abolish regulatory animal experiments. This was announced in 2016 as an ambition to, in 2025, no longer have any regulatory testing on animals. We are now much closer to 2025, and I don't know how realistic this is anymore, but it is a strong positioning, and these do have an impact. Another aspect of this, which I also want to mention, is that in 2015, the European legislation protecting animals used in research, so the directive I cited on the first slide, was challenged by a European Citizens' Initiative. And the European Citizens' Initiative is a mechanism that exists for European citizens to be able to, if a sufficient number of citizens is behind, to get the European Commission and the European Parliament to reconsider a piece of legislation. And the Stop Vivisection initiative was one of the first citizens' initiative to be successful in gathering enough signatures, and this was in 2015. And their demand was basically to revoke the Directive 2010/63 EU and replace it with a much more restrictive directive that would phase out biomedical research using animals, or the use of animals in biomedical research, by 2021, so by the present year. They were not successful. They were successful in being listened to and getting their initiative considered. They were not successful in bringing about the changes that they asked for. The response by the European Parliament and the European Commission was basically that we have this European directive. This is the fruit of very substantial, high quality, extensive work to develop the best legislation in the world. And if we make it illegal to use animals in research, this research would go somewhere else where the animals are less protected. So all in all, it would not be. It would be worse if we restrict the use of animals in research. So basically, this was a kind of a checkpoint for where the European legislation is. The response by the legislature is we still consider it necessary to use animals, but we also still work towards this goal of full replacement. And the European Commission is really using this replacement as a central theme in their outreach activities. There have been 2 conferences now, one in 2016 and one earlier this year, which are all strongly focused on the replacement of animals, and not only in testing, but also in a biomedical research perspective. So what I think and what I hope is happening now, and what I think we need to be happening now, is a shift in the focus from finding non-animal alternatives, which is what happened in the cosmetics testing process, to optimizing biomedical research models. In toxicology and safety testing, it's a question of standard methods for standard purposes, and this being highly regulated. So it's any normally company that is engaged in this activity have to use standard methods that are set, standards that are set not by themselves, but that are set by regulatory agencies. So these regulatory agencies have a very important role. If we think about research, this is a completely different context. Diverse methods are used for diverse purposes, and there is little regulation. There is regulation obviously protecting the animals, but any researcher is free to choose whatever methods he or she find are the right ones to find out the answers to the question that they want to ask. So here we can no longer operate with a politically driven mechanism. Here we need a mechanism to be scientifically driven and to be focusing on improving the translation and the research potential of the models. This is important also from a policy perspective, because if we look at for what purpose animals are used in the EU, we see that in the latest statistics from 2017, 45% of animal use was for basic research. And if you do the exercise that I did for the first time in preparing this slide, and look back at the statistics that are available, and I went back to 2007 because that is the latest with EU. Well, I think now we are in EU 27, but anyway, it's after the expansion to the former Eastern European Union. So it's the same, more or less the same countries, and we see that proportionally. Basic research in 2017 represents a bigger share of the total use of animals than it did represent in 2007. Probably to a great extent because of the focus there been in testing of animals, so in safety, regulatory use, in moving away from animal use. We do see, although at a slower rate than I would like to see, that the EU-funded replacement research is shifting away from the super strong, almost exclusive focus on toxicology to being more inclusive. This review paper looked at Horizon 2020 research projects that work towards replacement. And there were at least 12 projects with biomedical relevance. And these were models using organoids, organoid chip, 3D models, and microfluidics. And this was without including the project that I represent in this context, which is the Remodel project, which is a project within the twinning, the widening research program of the Horizon 2020, and which is working towards developing advanced 3D models for biomedical research with a focus on organoid and organoid-like models. And this is a collaboration between I3S, which leads the consortium, and the universities of Utrecht, Copenhagen, and Würzburg, where we are drawing on the expertise in these 4 different institutions to work towards developing organoids and other advanced 3D models, and towards building capacity in our institution in I3S, a capacity that we also aim should be of use for the entire research community in, well, especially in Portugal. These are the kind of advanced in vitro models that we are speaking about. I borrowed this from a report from the European Commission Joint Research Centre. We've got 3D human tissue cultures, which is a move towards greater sophistication from the 2D tissue cultures that are the basic model that's been used for many decades already. The 3D tissue culture use scaffolds to create the 3 dimensions. Spheroids continue to work with the 3 dimensions, but now adding spontaneous cell aggregation, so no need of scaffolding. And organoids, again, these are 3D models which develop from stem cells and recapitulate basic features of the organ that they've derived from. And then we've got a microfluidic or organ on a chip systems in which microfabricated platforms are connected so that you have more complexity than just a simple organ. I've spoken a lot about this from a policy perspective, but obviously there is an important other reason to use non-animal models. There are translational advantages of using them. Being based on human cells, one hopes and aims that nothing is lost in translation between species. The in vitro approach is more easily scalable for high throughput approaches. And the fact that these are based on human cells also means that it's possible to create patient-specific models, which allows drug screening for personalized treatment. And this can be done, for instance, creating organoids from tumor cells from a patient, and then testing different drugs and see how this tumor responds to different drugs to select the best course of treatment. Very briefly, before we got started with Remodel, we also wanted to know what was the perspective and the perception among our colleagues in the institution, and to figure out what were key obstacles. And here, the group leaders at I3S, about 2.5 years ago, defined as the main obstacles to implement these models would be lack of theoretical knowledge, but above all, lack of practical support. And in this context, the vision that we have for the future with Remodel is to set up a system where we can give as comprehensive training and support for advanced non-animal research models as we presently do for animal models, and to integrate the two so that we can really, as an institution, as a scientific platform that we are building up, help work with researchers to help them define the right model approach for their research question, independently of whether or not it uses animals. That's not the central point. The central point is to find as Donald Ingber said in a very recent interview in Nature Reviews material, what the most important human relevant experiment is, and if there is an in vitro model available that's been validated, that is the model that we should be using. And with that approach, we also aim to do something about you see the gray spot that Portugal is on this map of the European 3R centers. With the expertise that we are building up together with the expertise we have on refinement of animal models to create an expertise center also in Portugal. Thank you very much for your attention. And if you're interested in following the Remodel project, here is our website and our Twitter handle. And this will be available on the slides that will be shared with you. Thank you.
Thank you very much, Dr. Anna Olsen, for your presentation. We will now move for the second presentation of this afternoon with Dr. Joslyn Dimaggio, and we'll have time for discussion and questions in the end.
Yeah, let me first say thank you very much for the opportunity. This is a very acute topic. I think Anna did a very good overview of the challenge, so it will be easier for me now to go through actually what we do in this Cogento.
I will just, sorry, Dr. Joslyn, I will just interrupt you for 1 second to do a brief presentation of your professional path. Sorry for that. So just to, sorry, just give me a moment. Yeah, so Dr. Jocelyne De Manchau did her PhD in France. She did her postdoc at the Columbia University and Harvard Medical School. Her scientific interests relate to the development and function of lymphocytes, with a focus on immune tolerance to organ and tissues in health and disease. She joined Instituto Gulbenkian Ciência in 1998, where she assumed various management responsibilities as well. And she endorsed one term of presidency of the Portuguese Society of Immunology and is funding member and part of the steering committee of Cogento. And now I will give the word to Dr. Jocelyne De Manchau. So thank you very much once again for sharing with us the work that is being done in this research infrastructure. Thank you.
Thank you very much. Sorry for this. And
no problem.
So I think thank you indeed for the opportunity, as I was saying, and it's really a pleasure to have listened to Anna explaining the context of of the reflection we must have in research and in preclinics as well. As a sort of introduction to the debate that we may have afterward. I put these slides, which is just an example, a bit cute, of similarity between human and mice in this case. Complex disease, actually, that here is manifested just by a superficial phenotype, but that is actually associated with more systemic matter, and that's called the white spotting syndrome. That could be in good part reproduced in mice, which is a single gene defect. But it was just a reminder that if we use model organism for health and disease, because health and disease is much more defined by the medical principle on the organism than on separated units of biological components. And the other, on the right, this is the drawing from Darwin, for those that will recognize it. And this is to say that this is because of common genetic ancestries that we think animal models could serve the development of knowledge and transfer of knowledge to medical science. The most popular model organism for research and development, and also for some aspect of clinical research, are the one represented here, C. elegans, which is just a worm after all, but from which we know so much, because we know the future of any single cell during development. We know every single gene, every single expression pattern. So this is what makes a very good model organism. It's tractable, but it also has an amount of knowledge that we already acquired. Melanogaster is a fly, and this is a fruit fly that is very much used in research as well, and is also amenable to a lot of experimentation, large number, like C. elegans, large screens. It's amenable to genetic manipulation also. And in the animals that are more concerned by what Anna has discussed before me, are vertebrates, actually. There's no ethical issues nowadays for invertebrates or for plants, for this matter, although this is all living organisms. So the ethical consideration starts with vertebrates. The zebrafish, as it's called, is a common model now in biology, notably for development, and mice, of course, and other rodents. And what I wrote down is examples of C. elegans, is what I'd explained, cell apoptosis. Longevity of the organism has been quite investigated there, and with relevance beyond this species. Flies have been used to understand what restrict tissue growth with great success, and also could be exported to other species. Fish is often used for questions of regeneration of tissues, of skin. And in this context, rodents are necessary because they represent all the complexity of the immune system that is not present in invertebrates, and also all the complexity of mammals, which is particular biological concepts that are unique to mammals and therefore to humans. Now, if we speak about preclinical research, which was the title of this session, actually most of what preclinical research is doing is to attempt to reduce compound attrition during the first in-human clinical trials. So that's a phase that is after the discovery and development, so clearly, and is a phase that precedes what will happen in humans in terms of clinical trials, phase 1, 2, 3, and is essential to funnel the candidate compounds. There is a lot of preconceived ideas of how to evaluate the success of preclinical tests. And actually, there was a fantastic study in 2015 that is published in Nature Reviews, Drug Discovery, where the analysis amazingly engaged 4 huge pharma companies that accepted to report what had happened for 10, 12 years for a total of 800 and more drugs that were supposed to be orally administered and were tested over this period. And what the paper revealed to us is at what stage a drug fails. That is represented here in blue is the percentage of failure at each step. So preclinical testing indeed failed 40% of the compounds during this time. But the greatest reason for failure were actually issues of toxicology, as Anna has mentioned already, which means that those preclinical testing save us a potential harmful outcome, us humans. What is revealed also in this study is that a large number of dropout or attrition from drugs is actually changes in the rationalization of the portfolio of those big pharma companies. 30% is huge if you compare to the previous one. And thereafter, after all, clinical safety in trials represents only 8% of the failure of the drugs. 8% is significant if the toxicity were very serious in humans, which had happened, of course. And there one may want to think how to improve indeed these conditions. But what really we have to retain from this is that the preclinical testing are definitely protecting us from unwanted events. Now, in this EMA, the European Medicine Agency review by experts trying to figure out how to improve indeed the funneling of drugs, what they discuss extensively the matter of the post-market safety monitoring, which is here represented as an iceberg, a bit humongous iceberg, but this is a slide from the EMA review, which we have all heard about very recently, for instance, with the episode of the vaccines for COVID. And this concerns the fact that once we go through all those steps, there is still what we call the real life testing. And this is testing in large scale in humans that we can call experimenting in humans, therefore. And one matter that we can come back during the debate is that we have very limited tools to model human population heterogeneity. And this is, I think, a concern for the future. We discuss this later. So in this context, what is COGENTO? COGENTO has this name to stand for Consortium of Genetically Tractable Organisms. It is an effort that bring together actually 4 institutions in biological and biomedical science: CEDOC, Champalimau Foundation, Gulbenkian Science, and AMM. So the 4 institutes are in the Lisbon area, which is practical. However, the ambition is, of course, to be useful for Portugal. So this is a distributed research infrastructure. It has entered the roadmap of research infrastructures, was elected, and therefore received some co-financing from state institutions. And our logo is derived from this. Actually, all this little drawing at the bottom represents Drosophila, zebrafish, and mice, which are the 3 model organisms we are focusing on. And the other drawings represent trainings, which is a big activity of the consortium, phenotyping through histopathologies, and also transgenesis that we could also call genetic engineering, if you want. So in a nutshell, this is a distributed research infrastructure that's made this challenge of synergize technology development across 3 organisms that are rarely handled together: mouse, zebrafish, and Drosophila, and provide state-of-the-art services for those 3 organisms. Integrating diversified expertise means that we are all institutes with very different research topics, covering a large domain of biological and biomedical relevance. So we do provide customized service for research and development, but also for preclinical research. This is based on a very large number of highly qualified scientists and technicians. More than 100 are involved in this activity. We rely on state-of-the-art facilities, and we definitely have a cutting-edge research surrounding that helps in decision-making. This infrastructure's got a maximum score in maturity analysis, so we hope it will be supported further, but for sure we want to maintain it anyway. So as the keywords that I used, and a bit in line with what was discussed before, model organisms research in development are very often integrating genetic engineering, very controlled housing conditions in line with a number of regulations, and apply the 3 R's that were discussed before: the replace, reduce, refine. The new possible scenario in research and development in this biology and biomedical science is the renewed interest and obvious understanding of the very huge importance of the microbiota on the host, and as well as all the subtle variation in metabolism that induce homeostatic disruptions, with consequence that we are already discovering. So Cogento, if you want, is a toolkit set and can be used as a suite for testing. So we provide consultancy and training, not only on the legislations, the 3R, the welfare, but also on the design of testing or projects. And we also provide competence assessment and project and severity monitoring, which helps again frame the use of animals in reasonable limits in what concerns welfare. And you can imagine here that we do import strains or line of mice, flies, fish. And this cleansing procedures is something that will get rid of unwanted pathogens and make sure that we work with controlled animals in terms of their burden of pathogens. We have a service to edit genes in the hosts, so transgenic or CRISPR, such that one could produce tailored models to the needs of the investigators. We host, so we have a good capacity for fish, flies, and mice, where investigators or companies could test their variables. And we have services of phenotyping. Histology is a big one with state-of-the-art equipment. Metabolism for suspicious imaging, and of course, many other services that are in our institutes that can be engrafted. The endpoint of such a project could be, for instance, the germ plus cryopreservation, which in other words means freezing sperm or embryos to stop producing animals that are not used and reduced, therefore, in the reduction concern. I will show you 3 examples of 4 examples actually of advanced capacity and high tech of Cogento. So this is here a drug screening platform that is applied to zebrafish larvae that is hosted at CEDOC. Larvae is interesting because they are small, and this is larva of zebrafish. They are small, and here we have a picture of a 96-well plate, standard material for laboratory researchers, where one can distribute larvae and therefore test compounds in a large scale and automatize the process. We can also automatize the process to the readout, which in this case is a motorized fluorescent microscope. There is novelties, and we are trying to catch with all novelties in animal in model organisms. One of them is this Danionella translucida, which is becoming a model organism in the sense that we are accumulating, the community is accumulating knowledge and also capacity to experiment on those animals. And these fish are transparent and very small, even as adults. So it allows doing new things in large scale on adult vertebrates. That will be interesting to follow. Recently, the Cogento is hosting a platform for metabolic and analysis of mice in this case, with equipment dedicated for that, designed to re-monitor every little steps of food intakes, water intakes, body weight, activities, and respiratory behavior as well, with very high precision. So what Cogento can do is design experiments or testing that requires measuring this type of parameters and hosting such experiments. For instance, nowadays, the effect of probiotics on metabolic rates is, no, that was the one here. Another type of advanced facility that we have for many years now is in a clinic called Germ-Free Facility, which is a place where one can have mice that are devoid of any macrobiota or macroflora, as it was called before. And they are in cozy conditions. They live well, they breathe well. And we can, in those animals, ask questions, for instance, on the role of the microbiota on xenobiotics metabolism, which is more and more realized as important in drug decay. This is associated with another facility that we call the gnotobiology facility, which in this case, you can have mice with a controlled microbiota. You can define exactly which microbe they are carrying and whether beneficial microbes in general is what one is looking for nowadays. And that's where the point of the probiotics was supposed to pop and did. Okay, so this was examples. Now I just want rapidly to tell you where Cogental is situated in the world through actually this germ-free and xenobiotic mice. We are related with a European infrastructure. It's called Infrafrontier that takes care of repertorying, distributing, storing, and creating mouse disease models, which itself is related with the transcontinental infrastructures called the International Mouse Phenotyping Consortium. This provides us with a lot of advanced expertise, networking, and of course, awareness of political development, for instance, and new regulation in the European scene. It is maybe interesting to also insist that Europe is regulating a lot, of course, what we do in biomedical science, but it's also putting in place a large number of structures or infrastructures that are there to facilitate the development of new drugs for medical applications. That includes, we don't need to go to the details of all of this, but this CORBEL is really a way to have brought together from collections of drugs to their testing in vitro and hopefully soon in more sophisticated in vitro assays, including 3D structures, as Anna was discussing, to animal models, but also based on huge computer capacity for system biology analysis, and so on and so forth. So there is really a good scenario here that one should profit from. And in some instances, like us, we could be ambassador to branch in. For the concerns that were mentioned before, and this directive from the EU that is not only speaking about replace, reduce, refine, but really transparencies and openness also of what's happening in laboratory that use animal for experiments. We have in Cogental actually a good group of a good working group that brings together the ORBEAs. ORBEAs are the Portuguese organ responsible pelo bem-estar dos animais, which in English actually corresponds to the Institutional Animal Care and Use Committee. And this working group that brings together a good number of people with expertise in animal science and in legal issues are also connected in Portugal with the minister, the national entity regulating the use of animal for experimentations, with the other societies. And this is a very dynamic area and group, which I think it's remarkable, and certainly the directive has actually propelled something there. There is other European structure that also help ensuring high standards. Nowadays, we have to follow very specific rules. So also when we report in vivo experiments, which is good because it allows testing reproducibility much more efficiently. There is structures that standardized screens in mice models. There are structures that also define the quality of preclinical data. So this is a very fertile ground nowadays. And again, one needs to find a way to profit from all the existing structures and integrate them in our activity, which is one of the objectives of Cogento. Now, in terms of users, Cogento, of course, serves mostly academia. But it also has and would like to participate in helping and providing support to biotechs and private sectors. And here is the representation of the 9 companies we've been working with so far, and it's ready to expand. At the end, also, I want to say that we have a website, it's called cogento.org, so relatively easy, with presentation of the history of this consortium, the services, the training opportunities that we provide, and various resources. We organize Cogent talks, as we call them, which could be informative according to your activities, and also have educative videos and virtual tours of a facility to participate also in public engagement. So I will stop here. Again, thanking, of course, the institutions that engage in this adventure, and Jean Crouch, who is our project manager, and he's carrying all of this, and more than, as I said, 100 actors contributing to this effort. Thank you for your attention.
Thank you very much, Dr. Joslyn Dimaggio, for these very comprehensive presentations. I'd say both very comprehensive presentations and complementary as well, in a not so common topic within the national context to be debated. So we will now move to the Q&A roundtable like part of the webinar. The idea is to discuss a bit the trends and the future of preclinical research globally. Of course, if you have any questions now, it's the time to share with us and with the rest of the audience in the Q&A box. So feel free to start asking your questions. In any case, I would start, and I would dare to say that probably much of what has been the preclinical research in the past couple of decades has changed a lot, and has brought new players to the game, novel challenges, and even more questions that must be put forward and discussed in fora like this one. So I think this is an important opportunity to discuss. If there are no questions, I would go for a first one, a more broad question for both the speakers here today, and would be, what is, in your perspective, of course, or have been the most important global trends in the past decade that you'd say have shaped the field of preclinical research? I don't know if Dr. Anna Olsen wants to start.
Yes, I can do that. I'd say, sorry. I'm not sure what's happening here, but we've got a few. There was a loud noise over a flying airplane. Sorry, I'd give 2 examples. Much more sophisticated genetic genetic modification, and I'm sure that Jocelyne can develop that technologically much more than I can, because I see them as they come to me as the head of the Orbea in my institution. So conditional knockouts, knock-ins, where you have much more control over when in the development that the genetic function that you want to control when it happens, and use of zebrafish. These are the two big changes that I see. There is one that I would like to see and that I don't think we have seen, but I think we can come back to that because you're going to. I think we'll approach the question of reproducibility a little later. So I'll leave that for the next step.
Okay, so Dr. Joslyn, if you want to jump into the discussion.
No, certainly. I will also. I would think that what has over the last 10 years, certainly the changes in the tools we have to introduce genetic modifications in animals is actually it's even enlarging the domain of the number of animal models we could use. The technique of the so-called CRISPR technique, the one that allows you to replace just a little piece of DNA, is very successful in many species, which is changing something, because before we were limited to a very few set of species where we could successfully introduce or replace DNA, and with CRISPR it is really enlarging. Of course, it means it's also enlarging the capacity to change genes in tissue cells, and to go with the organoids or to any other hope for therapies based on gene therapies, gene alteration. Not everything required. The success of the CRISPR, the impact what CRISPR will have on our life is way beyond animal models, of course. But that is changing our capacity. And actually, if we are very upstream of clinical concerns, it makes understanding of fundamental mechanism of biology in a very different manner. So that's clear. Well, I think what has changed is the regulation also, and we should probably mention. The presentation that Anna made was very good for that, to remind us the steps. It's not from yesterday, but it's taking there. And actually, it's not only the European Community bureaucrats, it's also the European Medical Agency is also referring to now the 3 Rs, which is quite new. And so this awareness that we should, this awareness of how to optimize, as Anna said, is really has been affecting positively, I would say. In that case, the biology and biomedical field. Really, the word preclinical research is something a bit difficult to define. I mean, I'm not sure it was exactly, in stricto sensu, it's really the drug testing. And I'm not sure this is the topic you wanted to focus on specifically or not. So drug testing, what has changed is what Anna says, is that we know much better also how to use stem cells and therefore make those artificial organs or tissues or structure or 3D structures as organoids, which of course are a part of the big problem of a full organism, but part for part, if we acknowledge this, it's going to be very useful. And that's also relatively recent, that we can do so much de-differentiation and redifferentiation. That's.
Mm-hmm. Yeah, thank you. So I would ask, I would ask Miguel if there are any questions from the audience also.
We have a couple of answers from the audience, and I think one follow-ups a little bit on these conversations on So, for instance, picking on organ on a chip, works on this field seems like to be quite outdated. A lot of research being done here, but the question from Hugo Sousa comes into how can we expect that the utility of actually these tools allow us to more fast translation of the data into what is early phase clinical trials. So how this is really supporting us or will support us even further to bridge this gap between what is preclinical research and what we expect to see on the early phase of clinical trials. It's what we try to predict or get information before going into humans. So I don't know what is your vision on this utility of making it fast, moving data into the clinical stages, and how this is being used already today or should be used in the future, organ on a chip.
My answer to that question is this will be dependent on the regulatory acceptance. What kind of evidence is required to approve first in human trials? And I think that is where the crucial one of the crucial challenges is, and I think we don't know at this moment. I think we will know. This question will start to be asked, and this will start to be challenging the present regulatory approach probably during the next 5, 6, 7 years. And at this point, I find it very difficult to make a prediction.
Yeah, discussing the time it takes is always painful because it takes a lot of time, but to transform a knowledge in a medical product, successful medical product. I really think that's what could be good is preliminary screens. Right? In general, when one has a compound that one wants to move to the clinic, one construct or one gather a collection of compounds a bit modified this way, that's why. So as first screens, the funneling may go faster. Whether it will go faster to first in humans, we will have to really balance the risk, of course, right? Of what is the objective. Now, in terms of, I think it will speed up discoveries and speed up screens. That I'm sure.
Okay, we also have here another question, also on these organ on chips, organoids, on how you foresee that this could be totally replacing the use of animals. If something that we can expect, or there is some areas or pathologies that you foresee that this replacement will not be expected. So in some way, is eventually the technology allow us to totally replace animals, or there is areas that we need to still rely on animals in the future?
So jumping into this question, I would ask also Dr. Anna Olsen, because she mentioned the advantages of these models, of the non-animal preclinical models, and I would ask her to comment a little bit on the major gaps and the state of the art right now, and how can or do you foresee that we can overcome those in the near future? Probably it's a bit futurology, but a couple of hints on this.
Well, this is really stretching my own competence in this field, but typically when we discuss this, one of the challenges that come up is, in a principal sense, the challenge is always when you need the system to be more complex and have more components that interact with each other, and that is an interaction that you cannot completely set up and model artificially, then the organoids as representing only one kind of organs have are limited. And a very typical example that come up and that I'm going to choose here because I know this is Jocelyn's area of expertise and it would be relevant to have her input, is the immune system. So when you expect there to be an immune reaction, you don't have that in a single type organoid.
Yeah, I think this is, thank you for bringing the immune part, the immune part, the neuro part, and now this microbiota part. We are, so biology was moving away from for breaking life in little units and trying to make it more organism centers and more back to physiological concept, let's say. And in the clinic, we all, because of this dilemma of we would like to lighten the use of animals, we are pushed to go back to fragmentation somehow. So I think it's a matter, that's where the discussion of the choices of models to test should be made out of an educated reason, a scientifically educated reason. If you are developing a drug that you want to break DNA, it's fine to test it first on cells, right? And will it break DNA? The 2D culture. Now, if it starts to be a drug that will solve issues of immune, the immune part is clear, right? The immune controls of tumors. Well, you have to have a system that is sophisticated to the point of mimicking the dynamic of lymphocytes, so the blood cells, their interactions with the non-lymphoid part of the hematopoiesis of the blood cells, and their interaction with the tumor, and their interactions with the matrix, and their way of penetrating tissues. So we may find very good and complex system in vitro, I'm not saying. But at one point, to place it in the frame of the organism, I don't know when we will not, when we will be able to spare us this. And we always, the question, the balance will be, which risk are we taking when we move it to humans? And we all, everybody's educated on the situation where drugs pass to humans and where and reveal other things. So it means that although the screen is quite efficient in avoiding catastrophe, we also all know that past the clinical stage 3 clinical trial, there is still unknown on the effect of the drug because of its combination with other factors that could not be included in the trial. So the reductionist approach is dangerous. And I know that's not what Anna is proposing. Anna is proposing non-reductionist in vitro system. And this is important to invest on that, absolutely. But we should always be keeping the perspective, or let's hope our regulators will keep the perspectives that a human body is complex with multiple inter-organ interactions, and touching one kidney may have consequences on the lung, and that will be difficult to model.
In this topic still, how do you see the role of technologies like AI in silico modeling in this context of managing the complexity, the risks, overcoming some of these limitations? So are these helping, or are they just adding another layer of complexity, actually?
Well, I can start to change, but they are helping. They are helping, but you know, AI for the time being, they produce information out of what we feed them. So we need to feed them with data that are relevant and unbiased, because if we feed them with biased data, they will end up with a bias and so on. So there is also a catch there, I would say, that if we reduce too much our production of knowledge, if we do not complete our production of knowledge, the AI will not help us more than a certain point. And so I do believe that system biology and modeling are helping us analyzing large set of data, which our mind are not very good at doing. And that's informative. That's help us make some predictions on notably on all those network structures, which is very difficult otherwise to see all the pin. It's like a pinball table and predicting where the ball will go. Well, after years of playing in the bar, you can get there, but it's what those system biology associated with AI are doing is to say, given all the network and the nodes and make it meta networks, you could predict that a disturbances here will have this consequence there. So this is a game that actually people, basic metabolics, I mean, biochemists people were doing years ago without the same equipment, of course. And we are reviving all of this, right? Which is good. And so it helps. Now, whether it will go to the point of having the matter of blood circulation pressure when assessing again the kidney structure, that will be, I'm not sure we are ready. We are not ready now. We may. And I think it comes to the sentence that Anna used, right? When it will be scientifically possible. So it has to be judged by scientists, I think, to which point we have saturated AI of relevant information. And I don't think we are there yet. And a small parenthesis is that each technological development revealed to us a new domain of ignorance that we had. And that's actually the macrobiota story is very fascinating for this. We all knew that we are carrying a lot of bacteria in our intestine, in our skin, in our ears. We knew that. But we didn't have the tool to analyze their complexity. And the tools were essentially new generation sequencing. So more the next of the new generation sequencing is what made us aware of how rich this environment is. And then you can start to dissect, of course, the interaction with the host. So technology is important. I think technology, yes, does reveal our ignorance in some instances. Sorry,
very, very interesting perspective. I totally agree. And I would like to hear from Dr. Anna Olson if she has any comments to add here, any ideas that she wants to share.
I don't have a lot to add to this, but more than that, this big data processing is obviously important in terms of the capacity that it gives us to process big data. Sorry that sounds circular. But it's not going to help us to formulate the intelligent questions. So it's the data that it's not only the quality of the data that we feed into the system, which is obviously very relevant, but it's also the quality of the questions that we ask the system and that we ask to the output from the system. Okay, and I think here the scientist's mind is still very important.
Thank you. So I would go now for the topic that we have approached or mentioned before about reproducibility. We know it's a major issue when it comes to preclinical research, to research in general, and as major repercussions to the subsequent phases, in this case in biomedical research, namely in drug discovery. So how can you improve it? How do you see this the recent developments and the way the research in this field is going to improve this topic? Dr. Anna Olson, if you want to start.
Yes, and I'm going to start by sharing one slide that is not mine, but that Professor Malcolm MacLeod from the University of Edinburgh has generously allowed me to use. Because I think it is, first of all, it's really very important that we understand what the problem is. And this paper is not new. This is, as you see, from 2008. This is from the Camarades Consortium, which do critical assessment of preclinical research in experimental stroke. And here they went through 11 publications, 29 experiments that studied that evaluated this particular substance from AstraZeneca, which has this code. And what we see on these 3 panels here is if the paper reporting the results also reported that the animals were randomized into treatment groups, that researchers were blinded to which treatment group the animals were part of when they induced a stroke, and whether researchers were blinded when they assessed the outcome. And what we see here on the y-axis for each of these 3 panels is the percentage of improvement. And it is very telling how much larger the percentage of improvement is when researchers do not report that they have been blinded. So if we don't implement good practice, and good practice here is randomization and blinding, we will overestimate the effect of whatever it is that we are testing. And this is really critical. I think open science, open data will help improving reproducibility, but the data is only as good as the data that went into the database to start with. And if good practice was not in place when the experiment was carried out, then sharing this data is not going to help us picking this up. So we need to be aware of this. And basically my take-home message here is good enough to publish doesn't really seem to be good enough, because peer review have accepted these approaches. This is all there in the public in the published papers.
Thank you very much for this input. I think they're very important messages. Dr. Joslyn, if you want to add.
I think the essence of the matter was said. This has to be, it relates also to education and formation. It relates to quality of the investigators. And therefore it relates to the agenda that is behind. And that is certainly an ethic issue in the behavior. And as you know, even in more fundamental research, irreproducibility is sometimes associated to simple sloppiness. Sloppiness is created by the urge of publishing, because publishing is the way to survive, surviving is the way to progress. So, but now we are discussing more severe issues, which are when pharma have a responsibility that is even beyond this, right? Because in this, now this is about testing compounds. So research funded by pharma, shall we discuss it? I don't know. Rules, I think rules, regulations are certainly needed. There is that this data management program are in place, are discussed. Statisticians are certainly people that should be integrated in teams. Data analysts should be integrated in teams, and this is an effort that has to be made to integrate in biomedical research, preclinical research, the expertise that are required. And this is more than hands-on animal testing, but as well designing the study. So I think the point is that this is not something insoluble, because we know how to do it for it to be reproducible. So it's a matter of implementing, regulating, and educating at the time. quite sure as well.
Thank you. Yeah, I think there are a lot of issues that have been raised, or topics that have been raised from this discussion, and that have showed that there are many more ethical concerns that only those related to use or not to use animal models, and I think the improvement and the refinement of the animal models, it's probably the key question that we want to and will address, probably, if we still discuss this and have the right people to in the table to discuss this kind of issues. I don't know if any of our panelists here today have any other comment to add to this session, because we're approaching the end.
If I may, I would like to add to the discussion of reproducibility. Lots of the responsibility obviously lies with the researchers and the research institutions where the research is done. But I also want to highlight a responsibility that lies with what is sometimes called the gatekeepers. So those that regulate, quote unquote, the access to the big fancy publications, those that accept, make sure that, or decide on what is published in the big shot journals. Because the more focus is on great promising results and the less focus is on how solid these results is, the more problems we will have with reproducibility. And part of the fault, I think, is of those who make this kind of decisions. There isn't enough attention and enough acceptance for the fact that results need to be solid, and that many early results perhaps are not.
Yeah, probably stop dividing results in positive or negative results would be a way to go. And yeah, I think this is a discussion that could go on and on for the afternoon, and it's probably a total change of mindset and the way progress is acknowledged and valued also in this field. So I don't know if Dr. Joslyn has anything to add to this topic or to this discussion.
No, it's going to be redundant. But again, I just want to say that we know how to make it reproducible. So it's a matter of fighting for this against the gatekeepers that are not only the journals, but also the journals do what they do because we subscribe to them and because our funding agencies delegate to them the classification of researchers and stuff. So the problem is very complex, but I think we know the tools to use to make reproducible science, and we know also how to test whether science is reproducible. So now it's to have the proper decisions made by the proper structures. Yes.
Thank you. Thank you for your inputs and for this very, very stimulating discussion. I think we need this kind of topics to be discussed and openly discussed more and more to have improved mechanisms and improved research as well. And this has important repercussions for the whole value chain of health. And of course, here in representation of the Health Cluster Portugal, there is this driver of producing more relevant and more robust science that, of course, will end up being of the most important value for the people and for the social development of the country. So thank you very much once again for participating in this webinar. I will close now this session. I will just share my screen to give you here and to leave you with the QR code where you can find the upcoming webinars of this series. So you can also follow us at LinkedIn or Twitter. And about next week, we'll be here at the same time, same place to discuss about the early health tech assessment. So hope to see you there. Have a nice weekend. Thank you very much.